established, one for established investigators who will be changing direction, and also for junior investigators who are launching their careers. Deadline is quickly approaching, January 30th. So please, if you're interested in pursuing one of those opportunities, we look forward to seeing your application. Also, we have summer research programs at each of the PKD RRCs. The links are there for different levels of students, either undergraduates or MDs, or training MDs who are looking for research possibilities. Look at each of those sites to see and submit names for people that might want to participate in the summer research experience. I want to also remind everybody that we do have a job posting on our website for postdocs or faculty positions, technical positions as well. Please, if you have openings available, we would appreciate if you put those up there. Take a look if you're interested in one of those type of opportunities. I also remind everybody that we are here for questions regarding just discussion regarding any of the reagents and resources that we have available that, can range from anywhere from, what are animal models useful for, reagents, what are the quality controls that go into that, experimental protocols, etc. So please reach out to us if you have questions regarding any of the resources that we have up on the site. And I would like to invite everybody to participate in our discussion board that's up there. It has been somewhat underutilized, and we would love to have a more robust discussion going there. And if you're on Twitter, we would appreciate if you guys would follow and retweet events and things that are coming out of the PKD RC. Also, and in light of one of the things we do is to keep everybody informed of ongoing opportunities and events that are happening in the PKD field. PKD Connect meeting is being organized with the PKD Foundation. I think it's Katerina Hopp and Michelle Patel are organizing this. It's going to be held in Denver, Colorado on June 23rd and 24th. They have a research component to that, so they're looking for submission of abstracts for that research meeting. The deadline for that is February 24th. So please consider submitting an abstract to that to make that a meaningful meeting. And so it's a pleasure for me to introduce Dr. Sun today. Dr. Sun is a professor in the Department of Mechanical Engineering, Institute of Nanotechnology at Johns Hopkins University. He will be telling us about kidney epithelial cells as active mechanobiological fluid pumps. So Dr. Sun, I'm going to turn it over to you, and thank you for participating in this.
Thank you, Brad. Thank you so much. Can you guys hear me well?
Yes, everything is good.
Okay, let me share my screen. Okay, yeah, it's a pleasure to be here to tell you about our work. As you can see, this is a project that's really a large collaboration between my group and the UMD PKD Center. So here are the key people that's involved. Iqbal Chaudhry here, he's a lead graduate student. He's graduating now. He's a postdoc at Caltech at the moment, but he led the project together with one of my students, now graduate student Morgan Benson. And we got tremendous amount of help from University of Maryland PKD Center, from Owen Woodward, Aaron Dixon. Without them, the project would not be possible. Okay, so I want to thank them in advance. Okay, so and the paper is published last year. You can check the paper for the technical details. There are lots of controls and other data that I'm not going to have time to show, but the publication's out there. Okay, so the critical concept that I would like to get across in this talk is the idea of mechanical fluid pump as it applied to the kidney epithelium. And everybody has water pumps in their houses, and they're characterized by flow rate, right? How fast the water can flow through the pump, usually measured in in a velocity unit, number of volume per time or something like this. But the other critical concept in a fluid pump is the idea of head pressure, meaning that if the outlet flow is at a different pressure, right? In this case, you can see that the outlet is higher, by some amount, and that means that there's a hydraulic pressure difference between the inlet and outlet. The flow rate will change. So if you're going to go buy a pump for your house, often you'll characterize them by what's called a pump performance curve. So this is a plot. So these are pump characteristics, right? It doesn't matter how the pump is actually built, but you can characterize them by measuring the flow rate as a function of the head pressure or the pressure difference between inlet and outlet, right? So if the pressure difference between inlet and outlet is 0, right, at the origin here, you expect the flow rate to be maximal, right? Because the pump is not operating with any kind of back pressure, so you have maximum flow rate. But if you raise the outlet pressure, right, then the pump flex will decline because there's a back pressure that's pushing back on the flow, and eventually you reach what's called a static head or the stall pressure. So that's the pressure at which the pump flex reaches 0, right? So this is, and if you buy different pumps, they will have different pump performance curves. They will look a little bit different, but conceptually, this is a little bit different from a filter, right? A filter, you expect the flux to be 0. So this will be the flux versus pressure curve for a filter. The flux is 0 at 0 pressure, and you need positive pressure at the inlet side, right? Inlet side before the pump in order to generate positive flux. So this is the sort of the major concept I'm trying to get across. And that's what we're going to show is how kidney cells potentially perform this mechanical function. Okay, so kidney cells are right in a kidney tubule typically. So lining the tubular wall, there's a lumen and there's flow down, and the fluid flux is going across epithelium from the lumen side to the interstitial side. So what we did simply is to unroll this epithelium into a microfluidic device, right? Putting a permeable membrane here, separating the upper chamber with the lower chamber. So kidney cells are sitting on this permeable membrane. There's an upper chamber here, and the lower chambers connected to an outlet. And then we control independently both the pressure and flows through the inlets here. So here you see that there are 4 inlets, 0 to 3. Typically, when we do experiment, we shut off the lower chamber, this particular inlet. So the fluid can only flow out at outlet 3, right? Upper chamber is, with the inlet 1 to 2, so we can flow fluid from 1 to 2. And then if kidney cells are transporting fluid from the upper chamber to the lower chamber, that can be characterized by this flux J3, which then leads to a displacement of fluid at the lower chamber, which then we can observe by eye, a rise in the fluid level in outlet 3. Okay, so this is actually done fairly simply with a ruler. So this is the picture of the device. You can see that the outlet side has a capillary that's connected to inlet 3, and then we can measure the fluid height as a function of time. And we know we can apply any number of, so all these pressure variables in the upper chamber, P1, P2 can be controlled, right? P3 is monitored, and we can change the flow rate or the shear stress in the upper chamber if you like. So by monitoring the fluid flux J3, right, that's rising in this capillary, we can actually measure the flux as a function of time and the pressure as a function of time simultaneously. And this device has been validated. We can show that if you grow the cells in here for a sufficient amount of time, you will form a tight epithelium with no leakage, and then only with sufficiently mature epithelium you will actually be able to see a pump action. And this device is actually designed with a number of perturbations in mind. You can apply fluid shear stress, as I mentioned. You can apply pressure easily. You can also change the media that's in the upper chamber independently of the lower chamber. So you can apply osmotic gradients. You can apply drugs and hormones to the system to look at how that changes the fluid flux. We can do lifestyle imaging immunofluorescence, as you'll see. Potentially, we could do a chemical analysis of the fluid that's passed through the epithelium. This we haven't done, but that's potentially possible because you expect the kidney cells to selectively, transport some of the chemicals that's in the upper chamber. And then we can also do transcriptome analysis, as you'll see also in our data. Okay, so here's a movie of the device that's with an epithelium, MDCK epithelium in this case, that's generating a fluid flux and creating hydrostatic pressure. Okay, and this is just done with a GoPro camera, very low tech. And then from this movie, you can extract the fluid height, right, in the capillary as a function of time. It looks something like this. And from here, right, you convert that into a flux, how much volume per unit time that's been passed as a function of the pressure difference. Okay, and it looks exactly as I described for a mechanical fluid pump. There is a maximal flux at zero pressure, but as pressure gets built up, the flux declines until you reach something that's close to a zero, vanishing flux. Okay, so we did a number of measurements in different conditions. We did multiple devices. You can do statistical analysis, compare different conditions, and what you see is that many things affect the pump performance curve. This is just one example showing if you change osmolarity of the fluid in the apical chamber, how does that impact the fluid flux? And typically we characterize the flux performance curve using two quantities. One is J0. This is the flux at vanishing pressure difference, right? When delta P is equal to 0, that's the maximal flux if you want. And then delta P star. Delta P star is the pressure difference at which the fluid flux goes to 0. So you can see that there's significant differences in J0 as well as delta P star if you apply osmotic perturbation, for example. We've also done other types of perturbations. Many things will actually change this pump performance curve. And then you can validate, for example, to look at how maturity affects, epithelial maturity affects this pump performance curve. And you can see that if the cells are growing, in this case, this is MDCK cells, if they're growing the device, as they become confluent, they don't immediately pump, right? It takes a little bit of time, a few days past confluency for them to mature, right? And then you can generate this pump flux, meaning non-zero flux at delta P equal to 0, right? If you're not sufficiently mature, your flux versus pressure curve just goes to 0, just like a filter, right? So, and then in the paper, we also have done additional validation, looking at dye permeation across the epithelium and so on. So the epithelium has to be mature, forming tight junctions, and then you will start seeing this pump action. And also, if you just grow other types of cells, for example, T3 fibroblasts, they never pump, right? The flux versus pressure curve always just goes to 0, like a filter, as I described to you earlier. Okay. And does the flux that we measure does it correspond to physiologically relevant values, right? And we can make an estimate, right? If you take just geometry of the nephron, surface area of the nephron, the total surface area of the nephron, and then the total absorption area of the nephron estimated, right, from human kidneys, and the fact that we typically can filter or transport 180 liters per day, you work it out, you get a number that's, that correspond to a flux about 1 to 10 microliters per minute per centimeter squared. So that's exactly within the range that we measure. Okay, so it seems to be giving physiologically relevant results. Okay. And I also want to mention that this, this microfluidic design is very similar to the Usain chamber, or the, Transwell type of assays that you probably are familiar with. Those things have been used for a long time to study kidney transport. But the important difference here is that we are quantitatively measuring the total fluid flux across the epithelium, while simultaneously measuring the hydrostatic pressure difference across the epithelium. Okay, so now that we established that kidney epithelia is a mechanical pump, what does that mean in terms of forces that are on the epithelium, right? So if there's a fluid transport that's happening as we're measuring, right? We saw that we're establishing a pressure difference across the epithelium. The basal pressure is greater than the apical pressure, right? So that's the delta P that's being plotted, right? So that says this pressure difference, right? This pressure jump is sustained, is applying a force on the epithelium, right? Because pressure is higher on the basal side, so pushing up. So, and that's sustained by mechanical tension in the epithelia, together with the permeable membrane that's attached to the epithelium, right? So the fact that it's pumping is generating a mechanical force in the opposite direction as the pumping flux. And this sort of is consistent with your own experience with mechanical fluid pumps, right? If you typically in a house, the pumps are bolted to the floor, but if you take a pump that's not fixed, right? If you increase the pump flux, you'll see that the pump actually feels the reaction force. It actually gets moved by the fluid transport forces as well. So that's exactly the same thing here. Okay, so we're observing this stall pressure, meaning that as a hydrostatic pressure is built up, fluid flux is changing, right? So one cell biological question we can ask is, what is responsible for stall pressure, right? Why is the flux decreasing with increasing pressure difference? And we know from decades of kidney research that NAK exchanger is one of the major driving forces of fluid flux, right? It's transporting sodium out of the kidney cell, and water follows sodium chloride, right? So that's one of the major driving forces of fluid flux that we observe. So indeed, if you add oil-bang, which blocks sodium-potassium exchanger action to our device, the pump flux decreases, right? We see significantly less fluid flux when we block sodium-potassium exchanger. But this still doesn't give us quite a clear picture, because the stall pressure we observe could be the result of the question is it an intrinsic limit on the action of sodium-potassium exchanger, or there's some additional active regulation that's generated by cells is causing this stall pressure. And this we can investigate using a tool that's developed by Mike Kaplan at Yale. He had this very nice system. He managed to develop the sodium-potassium ATPase with a snap tag, which then will bind this fluorescent dye TMR, right? So if you express snap tagged sodium-potassium exchanger in MDCK cells, you can, and then treat the cells with this red dye, basically, you can actually localize or visually observe location of sodium-potassium exchanger in the cell in real time. And that's what we did. So this is a movie of snap-tagged MDCK cells that's in our device. And you see that after you treat the cells with this dye, they will localize to the cortical basal lateral section of the cells, forming this nice cortical labeling. And this is in live cells, right? And then what we can do, because we can dynamically change pressure in our device, we can then monitor what happens to sodium-potassium exchanger localization as we change pressure. So in the first panel here, this is snap-tag sodium ATPase when the pressure difference is 0, right? There's no pressure difference across the epithelium. And then we can apply a 200 Pascal pressure at the basal side. And then we can also then remove this applied pressure, go back to the 0 Pascal, 0 pressure difference situation. So initially you see that we have very strong cortical localization of sodium-potassium exchanger, right? Once the pressure is applied, now you see a decline in the intensity, the TMR dye intensity at the cortex, okay, suggesting that sodium-potassium exchanger is leaving the cortex. And then when the pressure difference goes back to 0, we get restoration of the intensity of the cortex. And this can be quantified by looking at the intensity over time. It looks something like this, right? The intensity is kind of constant, and then apply pressure, it drops, and then pressure removed, it recovers. And if you look at a lifestyle movie of F-tractin, right? This is a lifestyle labeling of actin. You'll see that if zero pressure, you have this nice actin cortex at the basolateral side. And then if you apply pressure, okay, you see these imaginations, these ring-like imaginations. And then if you remove the pressure again, go back to zero, again, these ring-like imaginations will be restored. So here's another quantification. In other words, increasing pressure will increase these imaginations. And keep in mind that this is an acute pressure increase in the experiment. So sort of a step jump in pressure, right? And then I know that many of you in the field are probably interested in calcium, right? PKD is potentially a calcium channel. So we also looked at cytosolic calcium dynamics in DCK cells, right? So this is MDCK cells transiently expressing GCAM calcium sensor. And when pressure is equal to zero, you see that there's fluorescence and there's some flickering or some dynamics in calcium in the epithelium. But then if you add pressure, you get some dramatically increased intensity and flickering. So suggesting that pressure alters, right? Pressure alters the some calcium dynamics and cytosolic calcium dynamics in these cells, right? So one hypothesis is that if you apply pressure, cortical tension, membrane tension have changed to somehow allow the calcium channel to open, allowing inlet of calcium flux. And this is a quantification of MDCK cells expressing GCAM. Okay. All right, so those were all some of the preliminary results that we have on MDCK cells. One aspect of our device that we're pretty interested in is how it applies to human cells. And indeed, we can investigate how human kidney cells potentially also transport fluid. So, and this is with extensive help from Owen and Aaron from the UMD PKD Center. They gave us all these nice human cells from both normal kidney and also from ADPKD kidney. And these cells, human cells, in the primary cells, they grow nicely in the device, forming tight epithelium again. And then this is the movie showing the pump action of the different cell types. Now notice that for the ADPKD cells on the left here, okay, we're looking at fluid flux increase in the apical chamber. Okay, fluid actually has been passaged from the basal chamber into the apical chamber, whereas for the human cells, wild type cells, the fluid passage is from the apical chamber to the basal chamber, right? So there is a reversal of flux direction for the ADPKD cells. So this is a plot of the pump performance curve now for the wild type cells versus ADPKD cells. The ADPKD cells is plot with a negative pressure because now the fluid is being pumped from basal to the apical side, and then the wild type cell is pumping from apical to basal as usual. And also the stall pressure for ADPKD cells are significantly higher. Okay, and we also looked at mouse cells, in this case PKD2 knockout cells, again with the help of Aaron and Owen, right? So this is a doxycycline-based knockout, and that shows a reduction in PKD2 gene expression, right? And then if you put these cells into our device, interestingly, these cells do not show a reversal such as the mature ADPKD human cells that we showed earlier. The PKD2 knockout cells, because it's an acute knockout, right? That this is happening fairly quickly after the gene is deleted. We're seeing a reduction in the pump performance, okay, but not a reversal yet. And this is, again, a quantification of J0 and then delta P for these two cell types. Okay. And then the next thing I want to mention is that by applying pressure, we also saw a change in transcriptional activity in these kidney epithelia in our device. Okay, but different cell types, okay, seem to respond to pressure differently, right? So this is a comparison now with human cells versus ADPKD patient cells, right? I want to call your attention to ATP1A1. This is sodium potassium exchanger, right? If you apply pressure in normal cells, you get a lowering in expression of sodium potassium exchanger. Okay, that's consistent with what we saw earlier. At least they're leaving the cortex, right? But then in the ADPKD cells, you have a significantly you don't see a significant change. There seems to be altered pressure response in disease cells. And then here's a comparison between mouse cells, and the profile seems to be different, right? So somehow mouse is regulating fluid transport in a different way, and then their response to pressure change is also somewhat different. Okay, we haven't done a full transcriptional analysis yet, but by looking at the typical or the important genes involved in fluid transport, we already see an expression difference. Okay, and then finally, tolvaptan, right? PKD can be treated with tolvaptan, so it'd be interesting to see what it does, right? So what we see is that if you treat ADPKD cells with tolvaptan, you lower the fluid flux and the pressure in the device, right? So suggesting that now ADPKD cells it's generating less mechanical force or lower fluid flux, right? Consistent with the idea that you get smaller cysts in the kidney. All right, so this is sort of a set of results which is suggesting a mechanistic hypothesis for PKD progression. And this is just a hypothesis. We don't, many of the questions in here are not obviously fully demonstrated. But let's say that in normal kidney, you have fluid flux going from lumen to the interstitial in this way. And then, as I mentioned before, the restoring force in the opposite direction, right? So that's the force profile during fluid transport in normal kidney. And then if you have an ADPKD mutation, let's say you have one cell that's mutated, right? So this now have altered fluid transport, a lower fluid transport as in the knockout mice. Then you have an altered force profile, right? So the force is no longer uniform, or at least as a function of, where you are. Some regions may experience a slightly lower force. And that leads to a small mechanical deformation because now you have non-uniform force along the kidney tubule. And then the small mechanical deformation could lead to altered proliferation and the growth of the epithelium in response to this change. Now, I want to mention that when this happens, probably there's also altered flow and pressure profile in the lumen in response to this change in tubule geometry, right? So there is many variables are potentially changing. And we saw that cells respond to pressure in a very sensitive way. So the flow and pressure profiles are changed in this epithelium in the tubule, then many things probably will change in this progression. Okay, how much time do I have? Okay, I have a few more minutes.
About 3 or 4 minutes. Yeah.
So a few more minutes. I want to mention, this idea of fluid pumping is actually prevalent throughout physiology, not just in kidney. This is an example of a human embryo during initial bursting out the embryo, bursting out the zona pellucida. This is also occurring through fluid pumping. Okay, this is zebrafish, basically a sort of a gyroscopic sensing of, gyroscopic sensing of the zebrafish orientation. Right, this is occurring through this this organ, aortic vesicle, and then this aortic vesicle lumen also develops from fluid pumping. And then there's a connection between the fluid pumping mechanism and the actin myosin independent cell migration that's seen in some cancer cells. So there are many examples in literature that potentially connects to the similar type of mechanical action that I'm describing in the kidney epithelium. Moreover, and this is unpublished results, and we're still trying to generate more results, we're finding out that endothelial cells are fluid pumps as well. And they pump from basal to apical, right? In the opposite direction as kidney cells. And this sort of makes sense because, the blood vessel is right next to the, nephron tubule, right? The fluid coming out of the tubule has to go back into the blood vessel. So there should be a continuous flux of fluid going from the nephron tubule into the blood vessel. And you cannot, if you don't have a continuous flux of fluid, you have buildup of fluid, something will go wrong, right? So it makes sense. But what we're demonstrating is that HUVEC cells, this is again in the device, with no myoepithelial cells, right? This is just HUVEC cells themselves can generate fluid pressure from basal to apical side. We need more data in this area, but this is potentially very exciting. And then if you look at pressure profiles throughout the transport network that's in the human physiology, there are many areas where the question of pressure and fluid flow needs examination, right? So obviously the pressure difference in the large capillaries, large arteries and aorta, for example, are very high, right? And that's what's measured by blood pressure from your doctor. But if you go down to smaller capillaries and smaller veins, right, in the distal part of your physiology, the pressure becomes lower, it becomes comparable to the pressure differences that we're looking at, hundreds of pascals that we are reporting. And if this result is correct, that means fluid transport, active fluid transport is really occurring everywhere. And that should change how our view of fluid transport and physiology in general. And I just want to give you a systemic view, right? What we're currently thinking a little bit, this idea that, the network of fluid transport network that's in the body is very similar to fluid transport network that you find in a city, for example. And then you have pumps somewhere, you have pipes, right? You have potentially source of pressure and so forth, right? So this is a network, right? And you can write down the network diagram like this. It's very much like an electrical circuit, if you want. The analogy is almost complete. But the important thing to note is that the global circulatory flux, in other words, the flux through the complete network, is actually characterized by a systems curve. This is not dictated by individual pumps. The individual pumps contribute to the systems curve, but if you've ever had an electrical engineering course, you would know that the global electrical current through a circuit, in fact, is a complex result of everything interacting together, right? In a usual water pipe network, you're only looking at J and P, right? Fluid flux and pressure. In an electrical network, you're looking at current and voltage. Human physiology is potentially much more complicated, right? In addition to fluid flux and pressure, you have salt flux, sodium, potassium, chloride, glucose, so on. And then the corresponding variable for them will be the concentration gradients, right? So you have a much bigger space of flux versus potential, if you want, for human physiological transport. And why is this important? Because if you have a system curve looking like this, this is a systemic circulation flux versus pressure like this, and one pump in the network has a pump performance curve of this feature, then it's forced to operate at the intersection point. In other words, they will operate exactly at the intersection between the pump performance curve and the systems curve. Okay, a different pump, depending on what's pump performance curve, it will be at a different location in the flux versus pressure curve. Okay, if there is a systemic change in the overall circulation, system curve changes, let's say, for example, in a disease situation, now you see that all the pumps in the network must operate at a different point. They have to go to a different pressure versus flux region of the curve. And then why is this interesting? Well, if you look at a typical mechanical pump, this we don't know for cells, but if you look at a mechanical pump, if this is a flux versus pressure curve for mechanical fluid pump, there's another dimension that you should consider from mechanical design, which is the mechanical efficiency. Mechanical efficiency is not a monotonic function of pressure. In fact, it looks something like this. Typically, the efficiency is very low near delta P equals 0 or near stall pressure. There's an optimal point somewhere in the middle. Okay, so let's say that the system curve is such that you're near the optimal point, but now if there's a change in the system curve, you're forced to be operating at a different point along the system. You might be in a more inefficient point. This leads to cellular stress. Okay, mechanical fluid pump can break if they're not designed properly. They're sitting at a wrong region of the pump performance curve. So I think I will stop here. Here's some tentative conclusions, and I can open for questions.
Dr. Sun, thank you for a wonderful talk. There's some questions in the chat. Alan, I'm going to let you go ahead and start.
Sure. Very nice talk. Lovely. So in the tolvaptan experiment, are your cells bathed in serum, and is the assumption that there's circulating vasopressin? Because otherwise tolvaptan presumably wouldn't work.
Yeah, so in this experiment, we did not have vasopressin, but we did see a difference. So, I mean, I agree with you. This experiment can be done more systematically. Some of here is just sort of a preliminary result.
I guess my question is, if you didn't have serum in there, would tolvaptan still do anything?
Yeah, we haven't done that experiment. Okay.
Darren, I want to ask you a question.
Yeah, I'm just trying to understand this a little bit better. Nice talk, by the way. So you saw a difference in the directional flux of fluid by pumping between the normal and the cystic cells. I was wondering if you can induce the apical flow of fluid by treating the cells with cyclic AMP, the normal cells. And what happens if you use a CFTR inhibitor? Is all this being driven by the flux of osmolytes?
Yes. Yeah. So we haven't done the cyclic AMP experiment that you asked. We have done vasopressin experiment. If you add vasopressin, you'll get a different flux, an increase in flux, I think. Let me see. Here's some results. Yeah.
Okay.
So vasopressin treatment will change the flux. Yeah, and your question about osmolytes, yes, if you change the osmolarity, there's already an osmotic gradient across epithelium. Yes, so there's already naturally a driving force for fluid flux, right? So I agree with you. So, but we haven't dissected that carefully yet. I'm sure there's existing models or theories that can explain some of our results if we dig deeper. But yes, changing osmolytes will change the fluid flux. One thing I mentioned that we can potentially do, but we haven't done, right? So this is something we would like to do, is to actually analyze the fluid that's coming across the epithelium to look at if there's any chemical differences. If I change what's in the apical side, osmolarity or other type of composition, what will happen to the transport on the other side? So that's something that we can do. Just we haven't had the resource to do it yet.
Thank you.
Alan?
Yeah, sorry to hog this. You didn't mention much about the slope of J versus delta P. Is that representing the fluid resistance, or I guess in this case, the water permeability of the epithelia? I ask because here it looks like vasopressin actually increases the permeability, increases the slope.
Yes. Yes. So the slope changes if I change the stall pressure, right? Exactly why that is I cannot give you a full answer because this is the active pump, right? So it could be that there's more pumping action, or it could be that the permeability, permeability across the cell epithelium is different. This is something that can be investigated, I think. We haven't looked into the details of that so much. We have looked at, aquaporin expression, for example, that would change permeability, but permeability should not change the pumping flux in the sense that it should not change the active flux in the sense that it's driven by ion channel activity, if you want, right? So I tend to think that this change in all the changes that we're observing is because of ion channel activity is different.
I have a quick question too. The reversal that you see in the fluid transport in the human PKD.
Mm-hmm.
Have you been able to correlate that with changes in the sodium potassium ATPase localization or dynamics?
No, we haven't done that because it's more challenging to do the SNAP experiment, for example, in the primary cells. It's just a technical issue. Yeah.
And the other thing I wanted to ask is that, one of the models of cystogenesis out there is an obstruction model, such that a tubule becomes obstructed. Do you have any idea how that would be mimicked in this type of assay, or what that would do to the fluid transport mechanism?
Yeah, so I mean, I want to say that I'm not an expert on PKD in general, right? So I don't know all the hypotheses and the work that's been done on PKD, but at least I can tell you in our device, we can certainly engineer different obstructions to introduce pressure, non-uniform pressure. It's more complicated, but it's certainly possible. If what we say here is correct, meaning that, fluid transport is a function of pressure difference across epithelium, then if you introduce an obstruction, then there should be local pressure alterations. Locally, your pressure is no longer the same as, let's say, the background pressure, and that could lead to a change in the flux, right? Which then, of course, translate to a different force profile along the tubule. And then what happens downstream is clearly a mystery at this point, but any number of things could happen.
Abigail, you want to go ahead and ask your question?
Sure. Thank you so much for that talk. If we have time to talk, I just would love to see that slide again about the transcriptional differences between, the mouse and the human cells, and just thinking about sort of the implications of that, or any other thoughts you had about this. Thank you.
Yeah, so we see a difference. We see these cells respond to pressure application differently, but exactly what that means, I think it's
yet to be determined. Yeah,
right. It's not something we can explain at the moment. Yeah, maybe a full-scale RNA-seq will tell us something more. But we're just amazed by the fact that cells are so sensitive to pressure application.
Absolutely. Do you have an initial idea? Do you think this could be more about the difference between polycystin 1 or polycystin 2 mutations, or we just don't know yet, and we have to wait for more data?
Yeah, we just don't know yet.
Absolutely. Thank you.
These are patient cells with mixed genetic background. So
okay.
I cannot tell
you.
Sean, that was a lovely talk. I think you were going in that direction, but since the mouse cells are really monoclonal, and probably from the IMCD, have an IMCD origin, you might expect them to behave a lot differently than the more polyclonal human cells, and that may explain the transcriptional profile. I had a question about transport again. I think one of the favorite models is chloride secretion with CFTR on the apical membrane mediating the efflux of chloride and therefore water into the apical compartment.
Mhm.
I think the nature of the chloride channel is still under debate and review, but I just wonder if you had a chance to apply CFTR inhibitors in your model, particularly the human cells.
Yeah, we haven't we have not. There's such a long to-do list on this that yeah, this is one of the interesting items that's out there. My student graduated, so and then human cells are a little more challenging to work with, but yeah, that's something that we would like to also investigate.
Any other questions? You have a few minutes. All right, if there's no other questions, one of the things we always end with, Sean, is we reverse the situation here and we ask if there is something that the PKD Center could actually do that would help advance your research activity. I know you're already working with Owen in the Baltimore PKD Center, but is there? I would assume well-characterized cell lines would be something that would be very relevant for your types of studies. But are there other things that would help your research program that we could be working on?
Yeah, I mean, I think chemical analysis is one of the areas that we wonder about, and we have really no significant capability to carry out. So if there's something that's offered by your PKD center, that would be amazing to, give you the fluid that's transported and just tell me what's over it, what's in there. And then unfortunately, this type of work is a bit slow, as you can tell. So
you should try to make a mouse once. That's a bit slow.
yeah. So everything's challenging, I guess. Yeah, but we would like to get more people involved. Just more people looking at transport in this manner, and then I think any additional thing we find will be interesting.
Okay.
Yeah.
All right. Any final questions that anybody has? All right, Dr. Sun, I want to thank you again for a wonderful talk. It's a very interesting topic that I don't think nearly enough about. And
Thank you so much.
I look forward to the future of your research. Thank you.
Thank you guys for your help and support. Yeah.